The relationship between p-values and confidence intervals provides another perspective on hypothesis testing results. While p-values tell us about the probability of observing our data under the null hypothesis, confidence intervals give us a range of plausible values for the true population parameter. A 95% confidence interval, for example, tells us that if we were to repeat our study many times, about 95% of the calculated intervals would contain the true population value. This approach acknowledges the uncertainty inherent in statistical estimation and provides more nuanced information than a simple significant or not significant dichotomy. Recent criticisms of p-values have been further fueled by the replication crisis in science. Many studies with significant p-values have failed to replicate in subsequent research, casting doubt on the reliability of p-values as a measure of scientific truth. This has led to calls for more rigorous statistical practices and a reevaluation of how p-values are used in hypothesis testing. In response to these concerns, some journals have even banned the use of p-values in submitted articles. While this extreme measure is not widely adopted, it highlights the growing skepticism towards over-reliance on p-values in scientific research. Instead, many experts suggest a more balanced approach that combines p-values with other statistical measures and emphasizes the importance of replication studies. To address these issues, researchers are encouraged to adopt more comprehensive reporting practices. Instead of focusing solely on whether a result is statistically significant, they should report effect sizes, confidence intervals, and precise p-values. This approach allows for better interpretation of the results and acknowledges the uncertainty inherent in statistical analysis. While comprehensive reporting is crucial, the true test of hypothesis testing lies in the decisions we make. In the high-stakes world of scientific research and pharmaceutical development, a single statistical choice can determine whether a life-saving drug reaches the market or remains in the lab. The line between success and failure is razor thin, and the consequences of these decisions can impact millions of lives. At its core, the decision-making process in hypothesis testing involves determining whether to reject or fail to reject the null hypothesis based on the sample data collected. This process is particularly critical in fields like pharmaceuticals, where the outcomes of these decisions can have far-reaching effects on public health and safety. Consider a scenario where researchers are testing a new drug, drug B, against an established treatment, drug A. The null hypothesis might state that drug B is not more effective than drug A, while the alternative hypothesis posits that drug B is indeed more effective. The outcome of this test can have enormous implications. If the data suggests that drug B shows a statistically significant improvement over drug A, it may be approved for public use, potentially benefiting countless patients. However, if the statistical evidence is not strong enough to reject the null hypothesis, drug B might never make it to market, even if it could have helped some individuals. This decision-making process is fraught with potential pitfalls. Incorrect decisions based on hypothesis tests can lead to significant consequences. For instance, if a new drug is incorrectly deemed more effective than an established drug due to a statistical error, patients may be exposed to ineffective or potentially harmful treatments. Conversely, if a truly effective drug is not approved due to overly conservative statistical criteria, patients might be denied access to a beneficial treatment. To navigate these complex decisions, researchers must consider both statistical and practical significance. Statistical significance, typically determined by the p-value, indicates the probability of observing results as extreme as those in the sample data, assuming the null hypothesis is true. However, statistical significance alone does not always translate to meaningful real-world impact. Practical significance takes into account the actual effect size and its relevance in the context of the study. For example, in a clinical trial, a new treatment might show a statistically significant improvement over the control group, but if the actual improvement is minimal, such as a 1% increase in recovery rate, the practical significance may be questioned. This emphasizes the need for careful interpretation of results, especially in healthcare settings, where even small differences can have significant implications. In the realm of hypothesis testing, two types of errors can occur: type 1 and type 2 errors. A type 1 error happens when the null hypothesis is true but is incorrectly rejected. This is akin to a false positive, where a researcher concludes there is an effect when, in reality, there isn't one. On the other hand, a type 2 error occurs when the null hypothesis is false but is not rejected. This is similar to a false negative, where a researcher fails to detect an effect that actually exists. The significance level, alpha, plays a crucial role in managing the risk of type 1 errors. Commonly set at 0.05, this threshold indicates a 5% risk of rejecting a true null hypothesis. However, reducing the risk of type 1 errors often comes at the cost of increasing the risk of type 2 errors. This trade-off is particularly important in fields like medicine, where both types of errors can have serious consequences. Balancing these risks requires careful consideration of the specific context and potential outcomes of the study. In some cases, such as testing a new cancer treatment, researchers might be more willing to risk a type 1 error, falsely concluding the treatment is effective, than a type 2 error, missing an effective treatment. In other scenarios, such as testing the safety of a new food additive, researchers might be more concerned about type 1 errors, falsely concluding the additive is safe, and set a more stringent significance level. Domain knowledge plays a crucial role in interpreting the results of hypothesis tests and making informed decisions. Statistical analysis provides valuable insights, but it should not be the sole basis for decision-making. Experts in the field must consider the broader context, including previous research, theoretical frameworks, and practical implications. Welcome